Just like a quick announcement for those that grad students that are enrolled in the conservation sciences seminar. I'm technically the contact person for that. My name is Ken Kozak. For those of you that don't know me, I've been on leave. I just returned yesterday, so I'm reintegrating, getting up to speed. I will send out an email with some information in that, with so you can contact me and just reiterate what basically the expectations are for this seminar. And so with that, I'd like to introduce J.C. Buckner. Hopefully, at least some of you saw her Coleshorn lecture last night. She gave a really, I would say, fantastic talk on basically the role of diversity in science, and in particular, evolutionary biology. A very personal account and a very enlightening one, I think, as well. And today she's going to talk more about she introduced this a little bit, but she's going to talk more about her scientific work. She's currently an assistant professor at the University of Texas at Arlington. Also has an appointment with the Smithsonian. So she does collections-based research, so stuff that's near and dear to my heart and some of the others at FWCB. She's going to talk about ducks today, which a lot of conservation people like. Todd, I know, likes ducks. But from the perspective, kind of merging the perspectives now. She's going to talk about macroevolution and basically the evolution or convergent evolution of diving behaviors in ducks, and how many times this has evolved, and potentially, I'm assuming, what the underlying causes of that are, consequences for diversification. So with that, I will turn it over to JC. JC, welcome. We're looking forward to seeing a great talk.
Thank you so much. Can everybody hear me? Yep. I want to thank again Kathy for the invitation and for giving me the opportunity to co-show a lecture last night. The story I shared was very personal, and so it was super important to me, and I was really happy at how well it was received, and I was really moved. So I'm excited to now do the easier of the two talks, which is to just talk about my science, which I'm really passionate about. As mentioned, I'm going to be basically talking about convergent evolution in waterfowl and looking specifically at geometric morphometrics and what that tells us about how different species of ducks are adapting to different dietary needs over time. So some of you were at that lecture last night, so I won't do my typical introduction, which is usually really long. I'll just do something brief for those who weren't able to make the talk last night. I'm a macroevolutionary biologist, and I identify as a Black woman, and that has shaped my experience in STEM. And it's fully part of my identity as a scientist as well. In all of these group photos, you'll notice I'm the only Black person, sometimes the only woman. And while I won't get into the details of what that means today, I will say that experience has made me incredibly passionate about diversifying STEM, and especially ecology and evolution. And so I've been heavily engaging and celebrating and working to expand participation of historically excluded groups. And I've mostly been doing that work through service to nonprofits and scientific societies. So right now, I'm the faculty co-advisor for the SACNIS chapter at UTA. And if you don't know what SACNIS is, this is the Society for the Advancement of Chicanos, Hispanics, and Native Americans in Science. The name is very specific, but they've since increased their scope to true diversity in STEM. So they serve everybody who wants to be a scientist. What I like about SACNIS is that it really embraces this idea of bringing your whole authentic self to science and not checking who you are in the door, and really rejects the idea of objectivity and acknowledges how society impacts how science is done, what questions are asked, who has access, etc. So it also allows for different ways of knowing and acknowledges that diverse people have always constructed scientific knowledge in various ways. I'm also the Vice President of Communications for the nonprofit Natural History Museums. This is a growing group of Black professionals across the globe who are working in or affiliated with natural history collections. And we have an annual event that showcases and celebrates this community. One of our goals being to identify and collaborate with colleagues who are interested in broadening participation, particularly in these institutions, And what I have up here is just a few snippets from our recent museum exhibition that celebrates the work of Black people and their contributions to natural histories in time. So this will next be on display, as far as I know, at the American Museum of Natural History next October, which is also our Black and Natural History Museum. So all of that experience and those values from this diversity, equity, inclusion work have contributed to the way that I teach and how I mentor my students and how I collaborate with other biologists on research. And combined with my research interests, that has been the foundation of the EIDER Lab. If you don't love ducks as much as I do, EIDERs are a group of ducks that I'm obsessed with. And so that's the acronym for that. And oh, I'm gonna mention, this is the king eider, which is my favorite bird of all time. Okay, so in my lab, we're a diverse group of people who study diverse organisms. I have 2 PhD students right now. One of them, Anna Palma, over there in the far corner, is working on diversification and biogeography of grasses, which are chachalacas, curassows, and guans, if you're familiar with those birds from Central and South America. I have an undergraduate who's working on immunogenetics of squirrel monkeys. So really quickly, it's a really cute project because squirrel monkeys during the mating season, they gain a lot of weight, the males, and they look like little sumo wrestlers, and they fight each other to get access to females. So it's adorable. I love them so much. I have another PhD student, Yvonne, who works on biodiversity of southern Philippine lichens. Yes, lichens. I showed you we're all over the place. And so I also have a postbac and a postdoc who are doing a lot of the geometric morphometric work with waterfowl and also looking more into incorporating fossil species into that work and really trying to get a holistic picture of that evolutionary history. And finally, I have an undergrad who's working on population genetics in a species of parasitoid moth, which is partially responsible for the decimation of an endangered buck moth in upstate New York and Canada. I said this last night, parasitoid wasps, super cool. Google videos of them laying their eggs in all kinds of organisms like bursting out like the aliens. And for me, it's the same thing. They're totally based on parasitoid wasps. So they're really cool. So I have this awesome group of trainees that I'm really privileged to work with. They're brilliant, they're budding scientists. And I think there is a lot of diversity in the people and the work that my lab does. So our science and our practice is really heavily influenced by those different perspectives, and having this diversity of people, voices, cultures, etc., that are engaging with this kind of biodiversity research is super rewarding for me. I talked about this last night. I think it's really necessary that if we want to preserve biodiversity, we have to have a diversity of people who care about biodiversity, and so broadening participation in these kinds of sciences is super important. And so my love for diversity among biologists, which is what I talked about, is paralleled in my research where a fascination with organismal diversity is really what drew me to science in the first place. And so our research is really based on the recognition that diversity and variation are vital for life, and we want to understand how that diversity is generated, maintained, and lost over time. And so our central question, how are these patterns of biodiversity we observe generated and maintained? To understand the processes that contributed to this, we try to look at adaptive changes and phenotype and genotype at different taxonomic scales, largely within phylogenetic frameworks. And so the goal is to kind of provide some insight into these major questions. The most obvious for an evolutionary biologist is what is the temporal mode of diversification? I say vertebrate clades, but as you saw, we look at all kinds of things. But today I'll just be talking about birds. How do abiotic and biotic processes interact to drive diversification and create evolution? And finally, how does integrating data from both living and extinct species change our understanding of diversification processes? And so to do that research at the scale of whole orders and families of organisms requires a lot of resources. And as macro thinkers, we are often collections based in our work, and the sampling for these projects would not be feasible if it weren't for these collections and the cooperation of the collections managers and curators and all the volunteers who help to maintain them. So all the research I'll talk about today is collections-based. So in our lab, our research focuses on collecting integrated data sets that include molecular data as well as morphological data. And sometimes if we're lucky, we can get one or both of these things from fossils and incorporate those as well. We then build our phylogenetic frameworks to look at the type of mode of diversification, but also to use these to answer more questions about processes of diversification. And these are our main ones that we look at: historical biogeography, adaptive genetics, and functional trait evolution. And so I'll be talking about these latter two today with the waterfowl. So I do have some focus, a little bit. Our two main study groups are Flatirine, so Central and South American primates, and our second main study group is waterfowl, which I've already been obsessing over, so I'm just gonna move on with it. Actually, we study Gallman series. So if you don't know Crassidae, which is what one of my grad students is working on, they're actually galliforms, and so we focus on these two groups, which are sister to the Neo-Aves, which holds the majority of more than 10,000 named species of bird that are currently known to Western science. And as you can see from their phylogenetic position, understanding the evolutionary history of this group of birds is key for understanding early bird diversification. And so while research on galliform evolution is ongoing, macroevolutionary research on waterfowl has picked up only in the last few years. And that's despite several descriptions of early bird fossils being assigned to the crown and stem of this order, Anseriformes, as well as Galliform series as a whole. So for example, Asteriornis is a fossil species that was described back in 2020, all the way from Europe. It's one of the oldest known neornithines that's been described. It supports the hypothesis that there were terrestrial, small-bodied generalists that survived the Cretaceous Paleogene extinction. And it also suggests this broader or alternative biogeographic origin for birds, as previously the oldest known neornithine was Vagabus from Antarctica, which suggested a southern origin for birds. So now we kind of have this broader distribution. And so I bring that up because it is a stem member of this Galliform series clade that I'm talking about, and one of the oldest birds that we know. So today I'll focus on describing what we're doing to understand waterfowl macroevolution. There's nearly 180 living species of ducks, geese, swans, and screamers. And if you've never heard of screamer, you should totally YouTube it and listen to it because it sounds insane. But the two main reasons why I'm pretty obsessed with waterfowl as a study system is because they have a pretty cool fossil record for a group of birds. It's pretty extensive, considering how small birds typically are. Their hollow bones don't preserve very well, so they don't typically have extensive fossil records. But given their water-associated habits, they often get put into depositional environments, and so we do have quite a bit of fossils for birds. And then the second reason is because of convergence. I'm obsessed. Convergence is amazing. You're going to find out why today if you don't already know. So first, the fossils. Like I said, it's a relatively rich fossil record for a group of birds. These are some examples of extinct early waterfowl species that have been described. Some of these are well accepted as waterfowl, some of them are contested, and so that's something that our lab is collaborating with the Smithsonian on to kind of review this fossil record and let people know, like, what are the fossils we're confident in, what are the ones that are dubious, and which ones need more work and more description and things like that. Some of that record consists of subfossils, which is really fun because sometimes you can get DNA out of those. And so it was actually some of these recent extinctions that are represented by these subfossils that drew me to waterfowl in the first place. So I talked a little bit last night about some of the failures in my early dissertation ideas. And so as a grad student, I started working on this side project that was looking at these two extinct North American diving ducks that were characterized kind of by these relict coastal distributions on either side of the continent, that those are suspected to have gone extinct because of human exploitation. They basically got hunted and or eaten to death. And for those familiar with ducks, eiders are carnivores, so they don't taste that great. So we think what actually happened is that they got egg predated quite a bit, and so that's what reduced their population numbers. So back in the 80s, Livesey did some phylogenetic analyses using morphological characters and pointed to a relationship with the sea ducks for both of these extinct species. And sea ducks are one of a couple of groups of diving ducks, which I'll talk about. But a lot of you, because it's the first time I've been in a fish and wildlife conservation department, will probably know what the main modes of foraging are for waterfowl. So that's dabbling, diving, and grazing. But here's a quick clip, just in case anybody else doesn't know, to illustrate what I'll be talking about for Rainmaker's talk. Warning, the music is a little itchy, but okay, we're about to stick to the water. That's actually not good. Okay, so now you know the difference between divers and dabbling. Basically, dabblers, they feed on the surface, they upend their little butts, they stick out the water while they're feeding, whereas divers are fully submerging underwater to pursue their prey, whether it be benthic invertebrates or fish for some of the piscivores. So among waterfowl, there are multiple clades of these diving birds, and many of those morphological characters that Livesey used to place these extinct species are known to be convergent across those diving clades. So my advisor, Dave Jacobs, and I were interested in, corroborating or refuting these phylogenetic hypotheses about these extinct species. So I extracted some DNA from these subfossils of Candides, which is the older specimen, and some toe pads of the Labrador duck, which only went extinct in the late 1800s. So we have some skins from those birds. And we got near complete or complete mitochondrial genomes for both of those species, respectively. And we compared that to a lot of the mitochondrial data that was already published for waterfowl at the time, which included about 30 other whole mitogenomes across water. And so the morphological hypothesis is corroborated by the mitochondrial DNA for the Labrador duck. It's an eider, and specifically sister to cellular eider, which I have pictured on the slide over here. And so in this phylogeny here are the Mergini, which are the sea ducks that includes the eiders that I was talking about. And right here is Pemphirochus labradorius, that's right into that clade. However, we do not find this to be the case for Candides. We actually found that it didn't belong to any group of known diving ducks that we're aware of. So it's actually closely related to some of the more typical Anatini, which are groups like mallards and teals, which is this big red group here. And so you can see Candides is right, the sister group to this larger radiation of dabbling ducks, actually. So I was so stoked because I thought this was going to be the most boring paper I ever wrote. Like, yeah, Lucy was right, bones, they said it was an eider, it's an eider. But The broader implications of this phylogenetic result is that Conidates represents an additional example of convergence. So you already know there's a lot of convergence across this group, but now fossils are showing that it's even more common than what we knew before. And so on this phylogeny, I've kind of mapped the ancestral character states for foraging behavior, diving, dabbling, grazing. And we see the convergence on these diving characters, especially in the skeletons of these birds evolving multiple times across this group. And there are at least three additional clades that I hadn't sampled at the time that I made this tree back in grad school. So we're looking at this point at about 7 appearances of diving behavior and morphology in this group. Another interesting thing about Candides is the limb proportions. So what we have here is on, I still don't know what it is. on your right, this is Candides bones, and it's being compared to a modern waterfowl species here. So what you see at the top is that the wing bones are really, really, really tiny compared to the average size waterfowl species, but here the limb bones are enlarged relative to a typical size waterfowl species. What do y'all think that means?
They emphasize walking rather than flight.
Fly.
They did not fly at all. So they were probably flightless, right? Because these are some teeny tiny bones, right? On a honking ass bird. Like these are some big legs. So this is like a moose-sized bird with really tiny wings. Okay. And so you can see that a little bit better in this artist representation of Candides. This is the wings right here, right? You think he's getting up in the air with those little things? So we kind of see this coupling of wing reduction and diving behavior along with insular or coastal distributions across several extinct and living waterfowl, what we're referring to as this flightless coastal diver syndrome. And so we can see that in several of these examples, the wing proportions relative to the body size are quite different from what we see in a typical dabbling duck like this mallard, where you can see his wings are extending all the way back here versus these and these like shortened ones, these moderate wings on some of these other flightless or flight challenge species that we see in the fossil record. So, oops, that's all of the cues that I was supposed to go through. So, right, like I was saying, a lot of these species are kind of restricted to islands or to coastal regions where they're probably relying a lot more heavily on aquatic macroinvertebrate species for their diet. So what is hypothesized in morphology for diving birds is that small wings are good for diving, but there's a strong evolutionary constraint to maintain flight in these birds. So these constraints seem to be pushed to the limits by divers that maintain flight. So species like eiders and scoters who still fly, but they might have a little bit of trouble doing it. So let's see if this video betrayed me. Oh no, it didn't get here. I'm sorry. So this video is just going to show you what I mean about the trade-off between diving and flight with cool music. Okay. There. All right, so you get the idea, right? So you saw how the divers, they just lift themselves up like it's nothing, and that diver has to work his ass off to get some lift under those wings in order to get up into the air. And so this is a trade-off that I'm talking about between optimizing the body for flight versus optimizing the body for diving. We just submitted a grant to kind of investigate this a little further, what drivers might be responsible for this pattern or syndrome of traits, the flightlessness, the coastal distributions or island distributions, and this diving behavior. One of the things that we're really interested in looking at is the effect of ocean productivity and the distributions of this bird, and whether that has any relationship to how often we're seeing diving come up into the tree. To look at that hypothesis and other questions, we needed a better sample, more robust tree than what I had as a grad student. Then we could take this kind of deep dive into these other areas of research. So we started putting together a phylogenomic hypothesis using ultra-conserved elements, and now have the most taxonomically complete tree possible for waterfowl. And this includes some of the more recent extinct species that I was talking about that have molecular sequence data available. So we've included 4 of those extinct species so far. We're still collecting the data from the fossil side of things to finish the combined evidence dating for the divergence time estimation for this group. But in the meantime, to answer these questions, we've relied on the traditional nose calibration. So this is basically the result that we get. I won't go into a lot of details about waterfowl taxonomy because I know no one cares. But this is basically the maximum likelihood result that we get from IQ tree. And what you can see is I've highlighted all of these major subclades that exist within Amphibiformes. So we have the screamers as the outgroup, which is always what we find. And then we have magpie goose, which is this weird bird from Australia. I like weird birds. That wasn't a diss. There's your signal, which are all the whistling ducks, and then we have Oxyurini, which include one of our groups of divers that we like, and we have geese, sea ducks, and so on and so on. So over here is our new list of suggested taxonomic groupings for this group based on the UCE tree. And I can't take credit for that because I'm not a good taxonomist, because I find it not that interesting. But I work with Mike Sorensen, who's really into duck taxonomy, and so he came up with this new classification. And so these bolded groups are monotypic lineages that only include the one species. And the ones that are starred are ones that we're not sure should be their own grouping. Maybe we'll collapse those into other groups later as we get more information on those groups. But yeah, we pretty much kind of cleaned up, the taxonomic understanding of waterfowl for this. And so now that we have this tree, we're starting to really investigate these questions related to convergence and feeding ecology. Waterfowl seem to be quite labile ecologically. So as an example, there are multiple origins of grazers. This is just 2 examples, one being the Egyptian goose, which is in that green group to Germini, and the meanest bird we've ever met, the Canadian goose, who's in the true geese, right? Then we have some benthic trainers. So these are ducks that fully submerge in the water and feed in the silt and soil at the bottom of the water bodies. We have an example from the Asia, and then we have one of these Oxyuris species in different parts of the tree. And then we have things like macroinvertebrates that eat like mussels, clams, this kind of thing. So they're developing morphologies to be able to deal with that type of prey species. And we see those popping up across the tree multiple times as well. We've kind of just assumed up until this point that like these changes in ecology correspond to the changes we see in morphology, but we've never actually formally tested that idea. And so our first question has been really simple. Does this ecological convergence coincide with morphological convergence in a significant way? And so to do that, we're basically looking at geometric morphometrics of a couple different elements. For the time being, we focus on the skull and the hind limb. Because like I showed you with condyles, when you're a diver, that hind limb is your bread and butter. That's how you get your food. Our postdoc, Dr. Chatterjee, is the one who collected all the geometric morphologic data, and you can see his little setup at the Smithsonian. And what I think is really cute is I sat at that same desk as a postdoc and collected my data, so we got to sit in the same place. Sorry. So he basically did all the landmarking for these different elements for dozens and dozens and dozens of these specimens. And so these are the diet categories that he came up with to test relative to these morphometric data sets. So for his diet categories, he has the herbivores, which are largely herbivores. They eat plants, whether it's on land or like with the swans, they're up and eating benthic plants. We have dabblers and mixed feeders that are largely feeding from the water column. And then we have our divers, like I said, that fully submerge and either pursue fish or other macroinvertebrates in these water bodies. And so he also split them up into different foraging strategies. So actually, I realized it sounded like I was explaining foraging strategies on this page, but that's not what I was doing. I was just giving a general overview of what these guys are eating. So herbivores is plants, dabblers, mixed species. These are like different plant matter and microorganisms in the water column. And then these guys are eating things like macroinvertebrates and fish. And then the foraging strategy is related to how they get that food. So grazers are eating on land. You know, these surface feeders are just upending and getting their food out the water without diving. And then these submerged species are actually completely submerging underwater. So we looked at some OU models to see whether or not these morphological shape values that we get for the different elements correspond to these dietary groupings that Ray came up with. And so first we're looking at the skull. And so you see examples of the extreme ends of each of the PC axes that are summarizing the shape across these different elements. And so here we have blue are our herbivores, our terrestrial feeders. We have brown, which are like our sea ducks. These are the diving grasping groups. The red are our dabblers. And in the middle, overlapping with a lot of this stuff, is our diving strainers. Again, those are the ones that submerge, but are pretty much eating these particulate matters in the system. These straining species seem to display the most generalist morphology, overlapping with lots of different kinds of waterfowl, while these other groups kind of separate pretty well in trait space across this trait space. So our best fit model was this multi-peak OU model, each of those peaks corresponding to these different dietary groups that we identified. So just for illustration, to show you a couple of those different skulls and where they fall in the tree, we see convergence on the grazer skull, these more pointed beaks that are used to like pull grass and things up off of the terrestrial surface. We also see the convergence in these diving specialists. So we get these big, broad, kind of flat skulls with a lot of robustness in the beak for dealing with those macroinvertebrates. When we look at the hind limb elements, starting with this femur, again, we have over here, the grazers are in blue, pink are dabbling, and green are divers. And again, you see quite a clean separation across these different groups in the trait space. The main distinguishing feature being a curvature in the femur that we find a lot of the times in the diving species. With the previous taxes, we see again, there's a lot of overlap between grazers and dabblers, but a lot less overlap in terms of the entire trait space of these diving species. So we're seeing again specialization. The main thing coming out with those is the medial crest being enlarged in diving species. And so the significance of that, one of the most dramatic examples of medial crest elongation in birds is the loon, which I'm sure y'all know about loons, right? So that allows for a lot greater muscle attachment for these leg muscles that are producing all the power behind those strokes when they're swimming, right? And so we're seeing the same thing to a lesser degree happening in our waterfowl that are diving to pursue food. And then finally, we have our tarsal metatarsus, little foot bones. Again, clean separation across, will clean, qualified across these different foraging modes, with diving species being over here, our grazing species being over here, and our dabbers somewhere in the middle. That's the list. Okay. And so the answer to our question is yes. We do see that the ecological convergence pretty significantly mirrors morphological convergence across over time. And we also see that there's some significant covariation between different elements that we look at. Obviously, there's really strong integration across all the different hind limb elements because it has to work as a unit, right? But we also see to a slightly lesser extent some integration with the skull, which we're not really surprised because the entire organism has to function in cooperation to be able to pursue food, especially for these diving species that are performing prey pursuit, right? And so we're really excited about continuing this work to look at other elements and look at the degree of integration across the body and which elements are very well integrated with each other versus those that are kind of less part of the equation and just the trunk of the body and are not really active per se in these predatory behaviors or foraging, I should say, behaviors. Also, interestingly, we found that these specialist diets, we are considering grazers and divers to be specialists, whereas the dabblers represent a more generalist body plan based on how our morphometric space came out. They typically have the highest net evolutionary phenotypic evolutionary rates, with divers who are piscivores having the highest rates, followed by these grazing species here, and then finally the diving filter feeders. Sorry. Okay, so I'm gonna shift pretty dramatically to talking about some Bayesian models that are helping us to figure out a link between these phenotypes that we've been exploring and the underlying genotype. So Scott Edwards and a lot of his collaborators have been coming up with these phylogenetic genotype to phenotype methods where they leverage a phylogeny and a set of convergent traits to look at how they correspond to rates of evolution and molecular sequences compared to a trait of interest. So in the original model, this is phylo-HCC. As input, we give our phylogenetic hypothesis, and we also give the program a background rate of evolution, and that really usually comes from sites like fourfold degenerate sites, which means that the third codon position, no matter what you change it to in terms of a nucleotide, is the same amino acid. So that's assumed to be a neutral rate of evolution. And then we look at CNEs, these non-coding elements in the genome, to compare whether those rates, the rate of molecular evolution in those non-coding regions match these background or neutral rates, or if there's evidence for conserved rates, meaning that they're slower than the background rate, which means they're being conserved for some functional purpose probably, or whether we see evidence for acceleration. And if we see the acceleration, like in this example on our target lineages, which are the lineages that have the morphology or phenotype that we're interested in, we think that we take that as evidence that those may be associated with the morphotype that we see. Okay. And so in this set of nested models, basically, we take the data from the CNE after calculating those shifts in substitution rate, and if it's the null model, it means that we find no acceleration in any of these non-coding elements, right? If it's model 1, it means that we only find acceleration for that conserved element on the lineages that we care about. And if it's model 2, it means that it's random. Accelerations happen on any branch and it's not related to the trait that we're interested in, okay? So we use a version of this that has been updated in the last year or so called Follow ACCC. So in that first example, we're looking at discrete traits, right? We're just saying, this is a diver, this is a grazer. What non-coding elements are accelerated on these branches? And this one, we can directly input our shape data, those PC values that we get from our geometric metrics. And what it actually does instead is compare the fit of a model where the substitution rate in our molecular data and our phenotypic evolutionary rates are correlated versus a model where they are not correlated. And so we identify all the non-coding elements that are at the same time increasing the acceleration of the substitution rates in the same way as the phenotypic rates or decreasing them. So we can identify both, right? And we test those models using Bayes factors. And so this is some preliminary data from the waterfowl hind limb stuff that we did with Scott's former postdoc, Sabir Shakya. He also looked at some of these wing elements, the humerus and carpal metacarpus as well. And so what this graphic is showing is that we have this cutoff Bayes factor of 5. All of these are genes that were within a 10,000 base pair window of the non-coding elements we identified to have an uptick in evolutionary rates at the same time as we see an uptick in phenotypic rates. Okay. And so to explore an example a little more closely, we'll look at the femur, because for some weird reason I really like the femur. So we found 2 genes that I'll talk about that are active in insulin production and regulation. That's this IMS gene and this SORBS1 gene. And way back in the 50s, they did some research on these genes in chickens and showed that the insulin and insulin-like growth factors actually affect limb growth. And specifically, growth of the epiphysis or the shaft of the femur can contribute to bowing or curvature. And if you remember, I told you the main difference between our diamond species and our non-diamond species was this bowing of the femur, the curvature of the femur. So I think that this is really exciting. It's really cool. We obviously would have to do some kind of, evo devo to, validate that these genes are actually active in the way that we think they are, but we at least are narrowing down to some candidate genes that we can consider for those kinds of validation methods. And so I think this is promising to be able to leverage these convergent characters across phylogenies to get at these candidate regulatory regions, as well as some of these protein coding regions that might be active in creating the trait diversity that we're seeing across our groups of interest. So next, we want to do similar analyses looking at the skull, which is a much more complex skeletal feature. So we might have to break it down into units like the beak, the brain face, things like that. And we also want to look at other parts of the skeleton, like I mentioned, for integration, but also looking at whether these specific genes are affecting multiple skeletal elements at the same time, because we're seeing these changes happening tandem across the body. Okay. So with that, I'm just going to acknowledge all my collaborators and my lab group and the funding that we received to do this work. And I love you, I love you, I love you. And then I'll take any questions. I'm sorry.
Right, on your PCAs for the different working modes and dietary preferences, you showed the different groups and around concepts called we also have outliers. Can you talk about the outliers?
Yeah, I think most of the time the outliers are waders, because I talk about. So there's like the magpie blues and the streamers and stuff like that. Those are usually the outliers that came out.
Related, like, there's a lot of overlap.
Mhm.
between groups, those dots, they totally fall somewhere in characters in the middle too, or not necessarily.
The characters.
Well, by the PCA, there's overlap between groups.
Yeah.
Those kind of hard to classify into a group,
Oh, yeah, so,
something between the apple and the diamond.
yeah. So with waterfowl, they're kind of annoying because they have a tendency to have a broad diet breadth. So with some of those, they cross multiple categories, and so those are the ones that we refer to as mixed feeders. They also have some flexibility depending on what season it is, so they'll eat one thing heavily during the breeding season, they're different when they're eating in our wintering season. And so we see a little bit of noise across those groups, with the species that are, they have some of the specialist features of the agriculture grazers, but they still are pretty close in terms of overall body plans and dabbling, species.
So, I may not have determined it correctly, but when you see these cases of what you're calling convergence, looking at the rates of language substitution, of the genes, were those like across, those were across groups that convergently evolve the same ecomorph, if you will, they have
Yes.
the same cluster of genes that are basically have higher positive selection. So is that like, how do you see cases where there's a different set of genes that are upregulated? I guess I'm getting a question of whether you can distinguish parallelism in a sense. It seems like that would be a case of parallelism, all the same genes are being upregulated versus convergence. Can you get different genes producing?
Oh, yeah.
Same
Good
thing,
question. So with this particular method, we're not gonna be able to distinguish that because we're only comparing this model where everything's the same or things that we're identifying as convergent. Across the genome. So it would be a different exercise to look across the genome at what kinds of regulatory reasons and the legal reasons could be different genetic pathways for achieving the same phenotype. So we don't have to do that.
I mean, based on like how I was trained in the past, so like convergence would be like, there would be different sets of genes for something in the evolution of the same phenotype, versus parallelism would be.
There's-
same
there's-
turnout.
there's-
Yeah,
Sure.
so you have any sense, like, what do you, I mean, just where you have no sense of this parallelism or convergence in a sense?
I mean, it could be parallelism. I think it's early to say, which is why we're gonna get preliminary data. Our sample size should long have like 27 genomes across the tree. We're gonna increase that to 65 later. But, yeah, it could be parallelism because, you know, that same event that doesn't come from nowhere, their body plan is pretty restricted. And so it could be the combination of this kind of low morphological disparity across the group, but combined with high ecological ability, there's limited ways that you can vary the body plan, and so they might be using the same genetic copy that they can. Right, so it could, yeah.
What I just talked a little bit about that Devo angle, like, do you have any sense of like modularities, there's some modularity like involved in this, like those characters like evolves like rock, are they easily selected for because they're like a functional modulator? I guess I'm thinking of something with their bodies, work on the core of their modes of selection, and how actually the modularity of things like allows, you know, things to evolve more easily. I don't.
Yeah, I mean, so, yeah, of course, yeah, we saw on modularity and integration, and, I think we've evaluated all the elements independently when it comes to all the genetic stuff, and so we found different genes for different. So in terms of genetically, I don't know if we have any preference over the for integration versus modularity, but that's what we're looking at. In terms of more connected for the PCA. Concern.
If you had any same genes, that's it's giving a positive signal for different.
Thank you.
At your analysis and integration on your, the elements of the curriculum, but I'm wondering if you ever looked at elements we in there is integration, say, amongst the bill.
Yeah, we haven't done it yet. So, I was alluding to that a little bit with the genetic stuff, like comparing the baby soccer from the brain case and all that stuff, but we have to do the same thing for integration. But we haven't broken sales into multiple units yet, so yeah, I'm not sure.
When I was a grad student, there's another grad student who built a dive tank to study underwater foraging behavior. And that's really cool to see like ruddy ducks with forage tactile and substrate, lesser stomp for pursuit phase of predators underwater. I wonder if you looked at or considered things beyond skeletal morphology, like visual acuity underwater, or things that might segregate along that sort of axis.
Yeah, I think it was cool, those data. But before that, it was interesting. I think we started with skeletal models because it's really easy to go to questions and get on data. But yeah, I think things like what you're talking about, visual acuity, energetics, all of these things would be really interesting to look at different variants of diving that occur because they're not all doing the same kind of diving. So I think it is more nuanced questions about how these different modes of learning are involved.
Were your answers really different from other diving species?
And there's levels to it, yeah.
You had like, did you do any data analysis to find like what environmental conditions might cause different species, especially among like divers, to either have complete flightlessness or just be flight challenged, or maybe even just be like fairly standard in flight?
That's a good question. And I say that because there's so many other brands with that question. So, like, we don't have any answers yet, but that's our next step. So that's like the social diver thing that I was talking about, that's like the subject of our grants, whether or not we can leverage these very specific cases of extreme specialization and find out more about what environmental drivers or ecological drivers might be leading to this, very narrow niche space, also things to maybe the average area and 30 people can say. So,
Right, I'll keep an eye out.
yeah,
Has anyone contacted you about the extinction, resurrecting Labrador ducks, and what the best surrogates might be?
no.
Rural waterfowl are very, it's very common for them to hybridize, like they're one of the, when it comes to birds, they're like one of the most common ones. If you found any or looked into this at all, have you found any, like interesting things that happen when like a dabbler and diver might hybridize, or does that even really happen?
I have not looked at any hybrid anything, in my work, we kind of like clean minds. But, yeah, I think there's really, it can happen. You can have crosses across these different genera and protocols, but I think they're extremely rare. And I think hybridization happens a little more often and more closely related species of waterfowl, which tend to be within radiations that are doing the same thing in terms of foraging. But it's not unheard of that they will hybridize across weird generic lines. But I haven't looked at it.
Well, that's for you. Fantastic talk. Look forward to seeing what comes out of this. It's really awesome questions from Aaron. At least from my thinking. Thanks for a fantastic talk. So let's thank you. I don't know anything about that.
I can say great talk.
They probably fit together like. There's so many. This stuff.
Yeah.
Have a whole career.